Hello, my name is Nada Lukkahatai, an assistant professor at the Johns Hopkins School of Nursing. I would like to give you a brief overview of my research study on a predictive algorithm to identify genes that discriminate individuals with the Fibromyalgia Syndrome diagnosis from the healthy control. This study is a collaborative effort among several institutes. The Fibromyalgia syndrome is one of the most common chronic pain syndromes that affect an estimated 5 million people in the US and approximately 2% of the world population. Without the clear understanding of its etiologies, the diagnosis of this syndrome relied on the patient's self-report of the symptom. There has been no known definitive prognostic or diagnostic biomarker. This study used a novel predictive analytical algorithm to identifying a group of genes based on the differential expression that can discriminate individuals with the Fibromyalgia syndrome diagnosis using the 1990 or 2010 criteria from the healthy control, and identify the functional pathway of the genes that have high discriminatory ability. This is a secondary data analysis of our gene expression. We identified the expressions of the discriminatory genes using the 4-chain differential and Fisher ratio. The discriminatory accuracy was determined using the leave one out cross-validation. The functional network were described from the Ingenuity knowledge base. The results show 57 genes that were highly discriminatory for the fibromyalgia diagnosis. The combination of these genes with Fisher ratio higher than 1.45 provide a leave one out cross-validation accuracy of the fibromyalgia diagnosis for more than 85%. The functional network analysis showed that these genes were associated with 3 canonical pathways. The 2 highest accuracy genes are associated with the mitochondrial function, oxidative phosphorylation, and glutamate signaling. This is the first study that attempts to utilize a predictive algorithm using the 4-chain differential, Fisher ratio, and leave one out cross-validation to identify a list of genes that can discriminate individuals with the fibromyalgia diagnosis from the healthy control. We hope that this study finding will initiate the investigations, discussion, and development of the potential prognostic and diagnostic tools for the fibromyalgia syndrome. To conclude, the study provides an overview of a novel data analysis approach and discuss a potential functional pathway and their involvement with the prognosis and diagnosis of fibromyalgia syndrome. Thank you for your attention.